this research on behalf of the senior author, Katherine Thilges, who just successfully completed her master's research and defended it 2 weeks ago, but unfortunately, work commitment at Pioneer did not allow her to be here. The goals of her study are shown here. She wanted to cytologically and microscopically characterize an identified transposon with the name of TGM9 in the W4M soybean line that causes both male and female sterility. She wanted to determine the mechanism causing the sterility in both the anthers and the ovules, as well as to identify the stages in which the development stopped, and then finally to summarize these results with future work. To give you a brief background of this double sterile line, it was given the name ST8 gene, which was identified among the progeny of the W4M soybean line, which is characterized by variegated flower color. However, the W4 locus controls this flower pigmentation, and in the unstable mutable W4 allele, the color purple is restored. At the ST8W4 locus, there are 5 predicted genes that are present. One of them, Glycine 16G, codes for a helicase, and this is similar to the MER3, which has been identified in rice, Arabidopsis, and yeast. This gene produces a helicase protein which controls crossing over in the meiotic prophase 1 and maintains fertility in the cystins. However, the insertion of this transposable element TGM9 disrupts this function, reducing bivalent formation and, of course, causes abnormalities following it. Therefore, this transposon insertion is specifically gametophytic, and it leads to the double sterility. Katherine followed a very recent study that was presented in PLOS 1 in an allied lab on our Iowa State campus, in which they were able to identify the progeny that were either wild type or mutant. Her work utilized 4 seed packets, and she had 7 plantings from which she was able then to draw the material for her cytological and microscopic study. And you can see the numbers for the wild type over here as the number of plants, as well as the mutant line. Vegetatively, both the wild type and mutants look identical vegetatively. This was at 5 weeks post-planting, and those plants were grown in a controlled greenhouse down at Pioneer under both photoperiod and temperature given conditions. However, at the time of flowering and post-fertilization, the wild type produced the developing normal pods, as you can see in the left image, and on the right side there were no pods formed on the mutant line, even though you can see flowers here forming. Looking at the flower development of both the wild type and mutant, you can see that basically they look the same. The mutant reverts to the purple color, as is shown here in these 2 pictures. However, at the time of pollination and post-fertilization, which is internal, on the wild type side, you get the development of very young seeds, as you can see here. However, on the mutant side, the gynecium produces only ovules which do not develop any further, and this is about the same magnification as that one, so cessation of ovule development occurs early and does not allow for seed development. When one does a chromosome squashes of male myocytes from early prophase 1 to anaphase 1, this sequence occurs with the wild type, the upper tier, the mutant here, and then same down here. The big difference is quite visible at the metaphase stage, in which the normal shows 20 bivalents here, with chromosome number, of course, of 40. However, on the mutant side, there are many univalents formed. So you can assume at this point, when these separate, there will be an unequal segregation of the chromosomes into the following stages of meiosis and later. Acetocarmine staining also showed some differences. Again, a comparison from the sporogenous mass stage up here to the mature pollen stage down here. And everything appears to be somewhat normal for both the wild type and the mutant, all the way to the dyad stage, even though we know from the cytological work things have changed at this point. However, the tetrad stage shows the major differences, in which the wild type pollen, or wild type stage at the tetrad stage, only shows tetrads, whereas on the mutant side there was a combination of pentads, triads, and tetrads. And by the time pollen is formed on the wild type, normal pollen occurs engorged, whereas on the mutant side, the male cells, whether they're late microspore or early pollen, have a variety of different sizes. Most are not engorged, and many are collapsed. We followed a slightly different procedure in that we wanted to look at a large number of anthers and ovules during their developmental stages. So instead of going through the traditional fixing, embedding in paraffin and resin, and sectioning and post-staining, we used the confocal because the anthers and the ovules are small enough to allow for optical sectioning all the way through them. So the upper tier of images shows the wild type development from the sporogenous stage all the way to pollen. And you can see that the development looks basically normal, with tapedal cells here occurring all the way up to the late microspore stage, when they start to degenerate and are gone by the pollen stage. The lower tier of images shows the stages from the sporogenous to basically the pollen stage. When one does optical sectioning through this mutant tetrad stage, one can see the variation in the number of individual microspore products at the tetrad stage. Following that, upon release, the microspores do start to show some variable size changes, and some are collapsed, and again you can see this in optical sectioning. And at the end, the late microspores or early pollen are variable in terms of size, shape, and with many of them collapsed. Again, the pedum is still there at this late microspore stage, which indicates that, again, the process of abnormalities that are occurring are gametophytic. Looking at the ovules, again, using the same technique, and again, the aldehyde fixation, the aldehyde provides the autofluorescence, and the BAB compound, which is benzyl alcohol and benzoate, allows for the clearing and the optical sectioning. The upper tier of images, again, is the wild type, beginning at the primordial stage of the ovules, and going through the archiporial stage up through meiosis and into the formation of a young megagametophyte, which upon pollination and fertilization produces an embryo sac with a young embryo, which you can't really see in this image. And this is the seed, young seed. The lower tier is the development of the mutant line, and again, we can go all the way from the primordial stage, archosporial, through meiosis, but at this point in time, things apparently are become abnormal, as we showed cytologically, and you end up with an abnormal megagametophyte, which does not develop any further, collapses, and this remains a young, small ovule. To complete the story, we looked at the mature anthers and pollen grains of both the wild type and the mutant. The overall imaging shows that externally the anthers look the same, and the top tier one shows that upon dehiscence, the pollen grains appear to be normal, roughly of the same size. There can be variation to a small percentage, and when these are transferred to the stigma, as shown here, there's a massive amount of pollen grains, almost all of which are in the process of germinating. The normal pollen grains are represented by this image here, which shows the typical tricolporate condition and the normal architecture of the outer exeme. However, with the anthers of the mutant, when they begin to dehiss, you can see different size grains as well as collapsed ones. And when these are transferred onto the stigma, there is absolutely no germination occurring, which we would expect based on the previous results. The variation in these grains are shown in these three particular images. They are abnormal in shape. Some are partially collapsed at the colpi and totally collapsed. And some of them have very weird architecture, all representative of the abnormality of chromosome separation back at the time of meiosis. So to quickly recap what I've just presented on Katherine's behalf is that the plants vegetatively look the same. The abnormality and the cytological work shows a uneven distribution of bivalence and univalence that is similar to the MER3. The abnormalities on the male side of the anthers show that these abnormalities become apparent by the tetrad stage, and on the female side, a female megagametophyte ceases to develop any further. So, with this in mind, there are two future goals. One is to continue looking at the various mutants, fertility mutants that are coming out of this W4M line, and to look at compare their cellular and developmental effects, and also to possibly be able to find one of the mutants that can be genetically manipulated further to produce a stable male-sterile system for hybrid seed production. The authors would like to thank DuPont Pioneer for the funding of this study, providing some of the research facilities for supporting the graduate work of Katherine, and the microscopy facility I direct for providing the SEM. Finally, all of the authors would like to thank Dr. Reed G. Palmer, who died right at the beginning of this study, for supplying all of the seed and actually identifying this particular transposon system, and for his direction in the early aspects of the study. Thank you.
on and skip questions. Thank you very much. Please.